Hi. So it's a privilege to introduce the speaker of today's public lecture, Professor Daniel Fisher. So Daniel started his career at the Bell Labs, and he's currently David Starr Jordan Professor of Applied Physics and by courtesy of Biology and Bioengineering Department at Stanford University. He has worked on a wide range of problems in condensed matter and statistical physics, and is particularly well known for his seminal contributions to the physics of disordered systems, including spin classes, superconductivity, and fracture mechanics. And for the last decade or so, he has also been interested in developing a quantitative understanding of evolutionary dynamics. His other research interests include cellular biophysics and neuroscience. Daniel is a fellow of American Academy of Arts and Sciences and member of the US National Academy of Sciences. He has received many honors, including the very prestigious Unsaga Prize of American Physical Society. He has also been involved in arms control and energy policy. And here is the really interesting bit. Daniel dropped out of high school, obviously to do great things in life. So it's great to have Daniel here to deliver this public lecture.
Okay, well, it's very nice to be here, and I feel both honored and a bit humbled that many of you came out from Bangalore, this rather large distance, to hear me. So I have to start with an apology. I was rather confused about what or who the audience would be. And so I first thought it was public as in non-scientists at all. And then I thought that it was a lot of physicists who didn't know anything about biology. And then I learned that there were a lot of students here who were in biology and maybe were less comfortable with mathematical and things. So I'm worried that the talk in the end won't be appropriate for any of you, but I hope there will be bits of it that are. And particularly what my main goal is, which is really trying to ask questions rather than give answers, and I'll only give answers to a few things. Is there a way we can decrease the feedback or not use the mic? Because I have too much trouble hearing myself too much, and I have a loud enough voice, I think, can probably manage without. So, okay. So the basic laws of evolution have been known for 150 years, and there's overwhelming evidence for evolution, in spite of what some people claim. However, it's not really understood at all from sort of standards of physicists. In particular, the actual dynamics of evolution, the dynamical process itself, is hard to observe and to understand, and the quantitative aspects of evolution, which I'll talk about a lot, that very little is known, and often the question is not even asked. Okay. Now, in some places in the US, in textbooks, biology textbooks, there is a little bit inserted saying that evolution is a theory, not a fact. But this is in fact backwards. I would say that evolution is a fact, but far from a fully-fledged theory, at least by physicist standards. So this is now, I think, this was really intended for the general public part, the basic laws of evolution, the survival of the fittest, and then the part which gets de-emphasized, I think, in the public often as much. Nobody had a lot of parties for Mendel's 150th anniversary. They did for Darwin's, was the discreteness of the heredity, which is a really crucial part. And the origins of variation initially were thought, of course, to be primarily by sex. That was Mendel could check. And it was only a little bit after that the sort of spontaneous mutations that came in by De Vries and then Thomas Morgan really in a controlled way in the lab. But this is still more than 100 years ago, so the sort of basics were known then. The crucial then extra part was where the genetic information was stored, and that's the DNA, and this is now, whatever it is, 65 years ago, so it was before I was born, even if only barely. Okay. What about the overall evidence? Okay. So most of the evidence until recently really came from the ancestry, the fossils and all the modern species, and this was to construct family trees, the phylogenies. And then there are these rather lousy examples that are given, this Petbird moths example, which is kind of a boring one and apparently is not really true anyway. That's sort of some evolution on time scale of 100 years. Much more interesting ones are on time scales of tens of thousands of years by looking at DNA sequences and looking at changes in there. And this can give rise, as I'll show, to the phylogenies coming from the DNA rather than from the properties of the organisms. And also, particularly, say, for the antibiotics, for other things, the specific mutations which can give rise to some change in properties are known. Okay. The phylogeny, and this is one of a contemporary of Darwin's, so this is of course based on organismic features, probably the organisms, the interesting features of them. It has no dates on it whatsoever, and here you notice there is man up at the top, the sort of crowning achievement of evolution. Okay. What has made this a wonderful time to study evolution? Well, really, it's the revolution in DNA sequencing. Okay. So from when the first virus was sequenced, 5 kb, the humans in the early 2000s, this is now on a logarithmic scale the cost of sequencing. This is Moore's law for electronics, how fast they get for storage. And here was next generation sequencing, and this enormous drop here now. And so it went from having a few $1 billion for the first human genome to now you can sequence cells in your body accurately enough to tell whether they're your original cells or whether they've mutated or become cancerous, and you can do that for hundreds to $1,000. Okay? So now it's not only doing things like sequencing the genomes of some species, there's now much more detailed things one can do. You can sequence single cells and sequence from populations, where you look at a whole population together, sequence from that, and that of course gives you properties about the diversity in the population. And that's something which is tremendously informative, and particularly for microbial populations, and something I'll come back to. Okay. So one of the things that was early done from DNA sequencing before one got anywhere near to this time here, which was making the phylogeny of now more or less everything. And viruses on here, they're rather different, but everything on here, and actually this involved the discovery of these archaea too by Carl Woese, and this is really from comparing the most boring parts of the DNA possible, of the ribosomes that make the proteins from the messenger RNA. They exist in all the organisms, they're the most boring possible, so this is sort of exactly the opposite of what was done to get the original phylogenies, which were based on interesting properties of the organisms. Here it's now based on the boring properties of the DNA. And this is an example of the highly conserved proteins from that. Okay. So if you look at this tree, there's a little bit up here, which is the organisms that think they're most important on the planet. And this progression here, I thought this was only a useful joke in the US, but I learned recently that it's useful here too. So this is the ones that think the most important organisms are actually these ones up here, the cyanobacteria. So the cyanobacteria are the organisms which evolved to be able to do photosynthesis in a way that they break up water and enable them to be much, much more photosynthesis, and then what made the oxygen atmosphere. Right? If it wasn't cyanobacteria, we wouldn't be here. Complicated multicellular life probably wouldn't be here either. So they're absolutely the most crucial one. They can do fine without us. The opposite is certainly not true. Okay. Now, I was going to show a modern version of this with all the different phyla of bacteria, but it's so complicated with all these bits and things which are only known by some sequence taken from some obscure environment, and there's no known organism that actually corresponds to that I thought I'd do the simpler one, show the simpler one instead. Okay. So from the basic laws, anything can evolve, right? Antibiotic resistance can evolve very quickly. If you waited long enough, you could spontaneously get a cell from nothing. Right? That's of course ridiculously long, but somewhere in between those limits of things that happen very easily and that are unlikely to happen ever, for whatever that ever means, are things that can happen on different time scales. Okay. If you're a string theorist, as some of the audiences are, you care about the basic laws, and you can basically go home. Basically anything can evolve. After that, it's all details. Well, unfortunately, the details are the interesting parts. Okay, so there's a lot of things, a small number of things that seem to have only happened once. One of those is the cell. We don't know that it's happened more than once. There's certainly only one that seems to be the ancestors of one common one. Origins of metabolism, reproduction, we also really don't know about. Oxygen-producing photosynthesis, that's the cyanobacteria. That's a really crucial one. As far as we know, it only happened once. We don't know more than that. This is one where if I were the creator, if I believe in such a thing, and I had sort of planted a bit of seeds of life, and then I got bored, after 1 billion years and nothing much had happened, and so I thought, okay, I'm going to allow myself one little extra fiddle, and I would make a little inorganic fiddle, and because the crucial cluster which does this is a calcium manganese oxygen cluster, and then the oxygen would come, and then things would get interesting again. So this is one we've no idea whether we were very lucky that it happened or whether it was sort of inevitable. But once it happened, it was sort of like silicon for computers, right? Is that when silicon's there, all kinds of other people have ideas and develop things that are going to be better than silicon, but silicon's so ingrained it keeps getting better and better that nothing else can invade. Right? And so the same could have been true here, that anything else that could have invaded, okay, the cyanobacteria already did it very well, no one is going to invade. And a lot of evidence for that is the algae in plants, which basically enslaved the cyanobacteria and made chloroplasts, and they, rather than reinventing it for themselves, okay, re-involving it for themselves. Okay. But what about things that have occurred multiple times? Well, a lot of things that one thinks of as being the most impressive, multicellular organisms many, many times. Eyes, well, at least 10 times, and I think probably this list is longer than this now. Okay. Now, intelligence, this one's controversial, depends what you mean. If you sort of mean borderline of higher intelligence, then there's apes, dolphins, parrots, octopus, actually ravens as well. Octopus is extremely far from us. Right, it's not even a vertebrate. These ones are pretty far, and they all got to this almost higher intelligence completely separately. Okay. Now, whether or not one thinks that means that, say, this is very close to being a human is controversial. I happen to think it is, that the extra step that would sort of be needed to push a chimp to be intelligent like us is not a very big one. Okay. Well, once we talk about things that have occurred many times, then we can start talking about probabilities of those. We can't really do that for things that have only occurred once. So we can start asking you, how long would they sort of typically take? Like to become multicellular, how long does that sort of typically take from some single cell organism? The antibiotic resistance, new species, right? Why a new species sort of million-year timescales? Major innovations like whole body plans and things like that. Okay. So a general question then is, can one sort of understand the dynamics of the evolution? What are the timescales of it depend on? Can one make any testable quantitative predictions? And I'm really going to mean any, okay, even really, really simple ones. Okay. So who cares about quantitative understanding? Well, I can give you a lot of examples of people who don't. Richard Dawkins, one of the popularizers of evolution, and he was writing about a paper saying that Darwin, about something which Darwin had worried about, of what's use of half an eye, about the amazing evolution of a vertebrate eye. Okay. And there was a paper claiming to sort of solve this problem and estimate it as, easily you would get it in 100 million years. And then I'll just read this. This is a criticism. Speaking as a physicist, I cannot believe there's been enough time for an organism as complicated as the eye to have evolved from nothing. Both questions stem from what I have unkindly called the argument from personal incredulity. Audiences nevertheless appreciate an answer, and I've usually fallen back on the sheer magnitude of geological time. Okay. So I call this argument for personal innumeracy. He has no idea what numbers matter, what don't. And the fact that he cites this paper as being a key one, when one actually looks and tries to understand, it is completely ridiculous. It has nothing to do with the question whatsoever. Okay. So it doesn't even get the question right. Okay. So what about the actual evolutionary timescales? Okay. Well, they seem to be really long. They're billions of years. But if you look on a sort of scale here, 1 second is human lifetimes, 1 year is the age of life. Right, so that's not that big a range of timescales here. Okay? And you actually notice a lot of things are rather compressed on this logarithmic scale that I've shown. In particular, human-chimp separation a few million years is only 100 times shorter than of all of multicellular life. Okay, so there's not very big ranges on here. So somehow, in some sense, things happen in very short time. Okay? The other thing to remember about this is all of this, these time information here came from physics and radiochemistry. Okay, so geology and radiochemistry, that's where the clock came from. Okay, so when I first started thinking about evolution, and I wanted to find out what people thought about numbers, and so I asked some evolutionary biologists that I met, several. I said, supposing the geologists and the geophysicists and the radiochemists got together and they realized they'd been using the wrong units. Okay, and that really, instead of life being of order 10 to the 10th years old, okay, it was really 10 to the 100th years old. Would it bother any of them? And the honest one said, no, we don't understand evolutionary timescales on the difference between 10 to the 10th years and 10 to the 100th years. Okay, and I think that's basically correct, certainly for the very rare things we don't know. I said, okay, what about 10 to the 10 to the 10? Okay, well then it was too big a number to think of. That starts being, if you estimate getting cells from nothing, you start having to put powers of 10 on top of each other, and it's hard to know what exactly one means there. So really the understanding of this and what these measurements are is from this. Only very recently can one have these biological clocks, which are extremely inaccurate. They're so inaccurate they can't tell the difference between this and this as far as the sort of timing of things. Okay? So this is the ages here with no biological understanding to them. So here I have a couple slides that were really aimed for when this, for some earlier talk I'd given, which for the public, which is a general question of what sort of determines the rate of evolution, even in really simple circumstances. So what's usually emphasized, and Darwin worried about this, he worried when he was told that Earth was only a few tens of millions of years old, he thought that wasn't enough time for selection to have acted. So he actually thought about these quantitative things, I think, more than a lot of his successors did. So this is then, you've already got something which is different, and then that grows in the population. I have a yellow lab, a lot of people like these chocolate labs. So this is now the reproductive fitness, it's growing in time. Okay. And there's plenty of time then in generations for even things would have slight benefit. Okay. But of course, to get this, I need to have the initial mutation to get that, and I can have a period without much, and then suddenly this comes in and can take over if these chocolate labs are popular. But this novel mutation, of course, can take a very long time to occur because mutation rates are typically very small. Okay. But what happens if I now go to a big population? If I go to a very big population, then the total number of mutations that occur each generation is the population size times the mutation rate. Okay. So this number could be pretty big, actually could be very big, even if the mutation rates are very small. Okay, so what happens here is, in fact, right away I basically get mutations, and then these take off here, and then when this takes off is basically limited by the mutation and then the selection. It's sort of some mixture between the two. Okay, so what's even in this simple situation is going to limit what happens depends on things like the size of the population, and we'd like to understand that in slightly more complicated situations. So what sort of limits the rate of evolution? Is it selection? That's what Darwin had thought. Is it mutation, particularly producing something which is interesting and novel? Right? And there's the whole questions in the evolution of the interplay between very rare events that maybe we were lucky to have happen 10 times in the on the whole Earth in the whole time that life's been here. Some other factors, things moving around, going to the right places, happening to be lucky when they've already evolved, and some combinations of these. Okay. So some of the puzzles, quantitative questions. This was a big puzzle when the human genome was sequenced. Why was 20,000 genes somehow enough to make as complex things as us? People had thought it would be 100,000. Well, I asked them, I said, why do you think 100,000 would have been enough? What was the intuition for that? And I never got a very good answer to that. 100,000 seemed a very small number to me as well. Okay. Sex, I mentioned something briefly at the end. The whole role of this, the drawbacks and advantages of it, is a subtle one and still one which I think very little is understood. What about the size of the Earth? Okay, so Dawkins said he falls back on the geological time. There's also geological size. So one way it matters is there's a very large number of organisms because the Earth's big, but the other one is how long it takes for mutations and new species and things to spread. Okay. Fitnesses. What is the meaning of fitness? Okay. The problem with fitness is that first it depends on all the environment, which depends on all the biology. Okay, that's one thing, but it also very much depends on the time scale. There's the short-term reproductive fitness, how many offspring you have, how many grandchildren, and so on. Then there's the intermediate term, which is avoiding extinction, right? And then there's the long-term one, which sometimes sort of called evolvability, which is that new species can come and diversity things from the same sort of group as you. So insects. Right, insects, any given insect species goes extinct in a pretty short time, but insects have been tremendously successful, right? So they're, of course, very fit in some overall sense. Okay? So this is really something which I think is very misleading. I hate using ohms for things, but this is one I'm in favor of because it does, if one uses fitnome, it does suggest it's a huge complicated thing, a complicated mess, and not just one thing. And I think getting away from fitness as if it were singular is important. Okay? The other crucial thing for understanding everything about evolution is this part here. We only see long-term successes. We only see things that have been anomaly successful. Otherwise, they wouldn't even get in the fossil record because it would be very small numbers. Most of them would just die out right away. So everything we see is sort of conditional upon having been very successful. Okay? And that means that actually certain things that seem as if they're impossible because they're kind of evolving for the future, and sex is one of those examples, this can sort of be a sort of way of trying to think about those, is things are going to look as if they're special. Right? The ones that did anomalously well, even with some amount of chance in it, they're going to look as if they were special and doing something special along the way. And that's a very general phenomenon. Okay. What about trying to quantify other things, the number of ways to evolve, routes to higher fitness? Well, I'm going to come back and say something about this right at the end. If you think of the ecology, what about number of ecological niches, like beetles, right? There are enormous kinds of beetles, and it said, okay, they all have different niches. But with bacteria, it's not at all clear what one means by that. Okay, so I talked a little bit about this yesterday. I'm not really going to say much about this today. Okay. So what are the difficulties? Well, of course, a general difficulty is that the number of levels of the evolution, the genetic changes make changes in the organisms. Those can change will determine how fast they grow. The change in populations changes the environment, changes the ecology, and there's feedback on this. That's a whole complicated process. Okay. DNA sequencing, it's fantastic, but we're completely overwhelmed by the number of changes, even on short term. So you see something, you sequence it, you've no idea which the important changes are, even when they're pretty small numbers. Experiments, and I'm going to mention some, they're hard, they're slow, they're difficult to generalize from, but they're really crucial to advance the understanding. Theory, one really wants quantitative questions, models, and a crucial bit is the generalization and extrapolation. You want to know what goes beyond the particular model. Okay? And then obviously a crucial bit is that theory, you at some point would like to have some connection to reality. You know, if one uses string theory standards, that can be pretty far in the future. We hope to do a little better than that. Okay. Now, it's very tempting to do computer simulations. Right? You make a model, you do it, you see some interesting things evolve, you make some nice graphics, and if you're good at your selling, you can get it on the cover of Nature. Okay? But the problem there is that the laws of evolution are absolutely general. So anything you do in a simulation can evolve as well. Right? You've no idea whether you've learned something from it. Something which is more general than a particular model, or things that you can extrapolate to the really big numbers that occur in nature, which I'm going to start showing. Okay, so simulations, they're important, but they're a very restricted use. There's a tendency, of course, to want people to do them, because in some sense they're easy, but they're very hard to learn things from. Okay. So if you want to understand evolution, you've got to learn to love bacteria. Okay. There's a lot of them around. You can do a lot of manipulations with them. They have tremendous diversity. Even in your mouth, you'll find tens of different phyla. The highest level, people claim this number here. I don't know where they pull it from. Somewhere I'd rather not be. And then at the low level, if you look just within one species, and this is again this particular cyanobacteria, there's a tremendous diversity within that. So that's a real problem and would like to try and to understand that. But all these things here means they're very nice for doing things in the experiment. They're very nice for sampling from the wild and trying to do at least genetic measurements from things from the wild, from DNA sequencing. Okay. Why is there so much diversity? Well, here we get into a problem. It's easy now to probe the genetic diversity. Right? The general rule in biology now is that anything that you can turn into a DNA sequencing measurement, you do that. So all kinds of things which even 15 years ago were done by different measures, now they're done by turning something into something you can sequence. Okay. However, probing the properties of the organisms is much harder. Okay, so you obviously want to connect these two, and we don't know how to do that. Okay. Colossal numbers. Okay. So the number of bacteria in you is about 10 to the 14th. That's more than your cells. Okay, so and they're quite important for you. Okay. The number of bacteria on the Earth, this is a pretty enormous number. Okay, somewhere in this 10 to the 29 to 10 to the 31. Okay. But now we can ask, how many bacteria have ever lived? So each time a bacteria divides, the genome is slightly different, so there's a new evolution experiment done with a new bacteria. Okay. So this is roughly this number times the number of generations, so it's some number in this ballpark with a big range of uncertainty. Okay. This number is really enormous. Okay. This number is enormous even by astrophysicists' standards. And the interesting point is, where does this number come from? Okay. This number is a product, say, this here, of about 10 to the 10th generations times 10 to the 29th individuals. Okay? So most of that number is coming from the fact that the Earth is big, that this number is big, rather than the time that life has been around as long. Okay. So if you think, as I do, that a lot of the interesting things in evolution, you have to be very lucky for them to happen, then it's the sort of total numbers that matter, and then selection can act on them. So it's producing the variation in the first place is the crucial part, either by mutations or by recombination from different organisms. Okay? So this is the important number, and it really is dominated by the age of the Earth, okay? By the size of the Earth, not its age. This, interestingly, is actually true for vertebrates. If you ask how many vertebrates had ever lived, how many evolution experiments we know of vertebrates, it's also dominated by the number of vertebrates alive at one time, rather than the amount of time that vertebrates have been here. And it's an amazingly big number as well. It's close to Avogadro's number, that one. It's not like this, but it's still very big. Okay. Sizes of genomes, they're in sort of megabase size. Mutation rates are very low. It's not understood why these are. Genetic exchange rates among bacteria, we have very little idea of them. I'll mention something briefly at the end about what we know about the consequences of them. Explorations of genome space. Okay. If I just look at a little bit of a domain of a protein, a functional domain of 20 amino acids, the number of possible domains is about 10 to the 39. Which means even every time I get a new bacteria, I'm exploring making new protein domains. I only will have explored all of those, certainly nowhere near having explored all possible proteins. Right? So there's a tremendous amount of redundancy. There's all kinds of different ways things could happen, and this is really a sense of it. The fact that even these little tiny segments of protein, that's about the similar numbers to this. Okay. We have no idea what numbers matter, or which combinations of these numbers. Okay. I'm going to say what happens in a particularly simple context. Even that is new and not trivial. Okay, and this is a combination about the rare events as to how big these numbers are. So there's one species of actually an archaea, which is believed to have numbers in this range. And supposing that in order for it to learn to evolve to eat something different, it had to make very specific changes. Changes to enzymes, to move a promoter so it can make a lot of the particular proteins to do this. And if each of those had a chance of like one mutation, 10 to the minus 9 each, probably all three of those happening in one generation, all three very specific events is 10 to the minus 27, which means in the whole ocean, it happens every generation. Okay? So extremely rare things can happen. Of course, in practice, there are going to be many different routes that it can get to do the same thing. And even more routes to doing something different, which might be better. Okay. So, now it's trying to get to something honest. Can one ever predict the rate of real evolution? Okay. So I'm going to do absolute simplest and most boring situation. Short-term evolution, what we can do in the lab. Asexual, constant environment, pure exponential growth, so there's no ecology. And in this case, the fitness is well defined. It's just the birth rate minus the death rate. No ecology, they just compete for the total food. I don't really even need that. Okay. Then we can talk about a sort of fitness landscape. This is an ideal physicist problem. Biologist friend, you say, here's the genome, here's its fitness, and then physicists, you just go away and think about that, okay, and try to work from that. What will it do? It'll take uphill steps in this fitness landscape. Those are the beneficial mutations. And we know from other experience that in any environment, even one the bacteria have seen most of the time, there are always many mutations available for them to do somewhat better in that environment. Okay. So a thing we can ask is, what is the speed of the evolution, the sort of rate of increase of the fitness of it? But then actually a much more interesting one can ask about is, what is the diversity in the population that gets produced by the evolution? Because that's something we can also go directly measure. Okay. Now the next few slides are really sort of intended for, the details of them are intended for from the physicists or those who are happy with the quantitative things. If you're not, then you can ignore them, but I at least want the pictures to be there. Because I wanted to at least do one honest model and some results from that. So this is the simplest landscape that one can think of. It's just a staircase. It's a staircase that goes on forever. And if you're on this step, you're fitter than the one on that, and you're fitter and so on as you go up. And then there are mutations. There's just beneficial mutations. I'm ignoring the other ones. And you go up here, there's a population, total population, which is held fixed by the competition. And we want to ask, say we start with everything down here, and we want to ask what happens. Okay. So what will happen is, first there'll be some mutation, and I'll get one individual here. Now that one will have descendants, they'll do better, so they'll grow. This one will die off, and then at some point I'll get a mutation to the second step. So I'll get the population sort of spread out in several steps, then I get one here, and it'll gradually go up. Okay. And I want to ask sort of, how fast does it go up this? Okay, and that's going to tell me the speed of the evolution is how fast I go up here. Okay, there's just 3 parameters. There's the total size of the population, there's the fitness, the mutation rate, and there's the steps, size of the steps in the fitness. Okay. So in a small population, it's quite simple. If I don't have many mutations each generation, then I have to wait till I get a mutation. I step up onto the next step. I guess I can go down and do this. I go on to the next step here. One goes up there. This population grows, and the whole population comes up here. Waits a while for the next mutation. That up here gradually goes up, comes up to here. Okay, and then I go like that. So it just basically goes up one step at a time. And in that case, you can say the speed is just going to be limited by how long it takes to find the mutation. So it's just product here of the production of the mutations. Okay. It's okay? It feels as if it's falling off, but okay. Okay. But what happens if I now have a big population? If I have a big population, I can produce many mutations each generation. So that means that before, this is a little harder for me to do, I've got this mutation has got up to here, okay? And I now have to think of me being like a spider, and before my body moves up there, another leg's gone up to here, okay? And then I start putting some weight on there, and then another leg goes up to this step here, okay? So I'm now spread out onto many steps, the population. Okay? So this is now spreading out on the steps. This is showing how big the population is on each step. And I'm actually showing this on a logarithmic scale. So this is the step which is the biggest occupied step. There's nothing above that. Okay? That's doing really well. It's fitter, it's growing fast. These ones are growing, dying out. But then the crucial thing, of course, is getting onto the next step. So that requires mutations, and that's a stochastic process here. So there's this process of getting to the next step. Once it's got there, those will start growing, and I'll eventually produce one to the next step, and so on. Okay. So to understand this, we have to sort of understand the balance between the selection acting here and the mutations acting here. And for the students in the class, I'm going to talk about this tomorrow, I guess. And so what is the result of this? Well, the result of this is rather surprising. Okay. The result of this only depends logarithmically on the population size and on the mutation rate. Okay. And you notice this combination n times n doesn't even appear. Okay, so the sort of intuition behind this is completely different. Well, I mean, there isn't any intuition, right? This is a model that's existed for a long time. These results are only a decade or so old. Okay, and this model is surprisingly subtle to understand, and we understand most of it now. Okay. So Michael Desai, after doing this theoretical work, he thought he would go off and do an experiment for a brief summer project, and went to Andrew Murray's lab. It turned into about 2 years, and he did a double experimental and theoretical thesis. He's now in a rather unusual situation, is that experimentalists think he's an experimentalist, theorists think he's a theorist. Usually people who do both, it's the other way around. Okay. So these were some experiments, and I don't want to explain these, but basically he made sure to keep things at pure exponential growth. He measured the speed of the increase of the fitness, the rate of increase of fitness, as a function of n and m over substantial ranges, and this is trying to look at it as a function of logarithm of this parameter. So this would be the traditional one step at a time, and this is what the theory was on the various experiments. Okay. Now, this verified the prediction. Okay. So if we were sensible about this, we would have just said, look, we've got this prediction, we've got this theory, great. For experiments, we've got great verification. However, it shouldn't. The model was completely ridiculous. This isn't like a staircase. The mutations don't have all the same size. There are complicated things going on. There are bad mutations and so on as well. Okay? So why should this agree at all? Well, that took quite a few extra years of trying to understand, but there's an important lesson in there is that the role of theory in biology is to be wrong, because that pushes you into other experiments. And some of those I'm not going to talk about in detail, but that was sort of a push from this. Okay? But the other thing is it makes one go back and scratch one's head and say, okay, do we expect for something to be have a chance of being better? And this is something which really comes from statistical physics, is the fact that when you've got very big numbers of things, certain features don't depend on the details, like properties of a gas or a solid and so on, and many more interesting cases. Okay? This we don't know if it's the case here, but we're starting to understand enough to know that it is with quite a wide range. So we now understand why this model, even though the model's completely wrong, the parameters don't directly mean anything, actually we should expect to connect to these idealized simple experiments. They're the most boring experiments ever done in evolution, until some other experiments I was involved in recently, which are even more boring. Okay? But they're very impressive. I won't show them, but they... Okay. What about connection to nature? So the theory is idealized continual evolution. I can keep going up the staircase, no interactions and so on, no sex. Is it useful for understanding anything? Okay. So the predictions, the interesting predictions are about the statistical dynamics of the genetic diversity, both the dynamics and snapshots. Here's an example of evolution of HIV evolution within one person. This is a phylogenetic tree, but this is got by sampling at lots of times. And so you can look at one time and look at the diversity. There's much better data on this now. If I tried to show this now, it wouldn't fit anywhere near on the slide. Okay. So this, it actually now looks like the statistics of HIV can be much better understood from predictions from this continual evolution theory than from existing theory, which was mainly sort of neutral evolution plus occasional selective sweeps. Okay. What's going on is that the HIV are fighting against each other the whole time. The human host doesn't notice that, it doesn't affect them. The HIVs are competing like crazy. Occasionally one of them does evolve something which will beat the immune system or do well against a drug, and then it affects the human as well. But the HIV, they're fighting with each other the whole time. Globally, and influenza also true. Also, this isn't nature, but other evolution experiments, and I want to show something from one of those. Okay. This is a simple evolution experiment done by Rich Lenski, E. coli with no ecology. What he finds, one of the first ones is sort of many routes to higher fitness defined from sequencing. You can look at which mutations come in, try to understand what pathways, what functions they do, and so on. Okay. But very recently, there's been, I'm sorry, I should have listed the authors on this. There's some measurements which they did of going back and looking at all the samples which he'd kept, and looking at all the mutations that had come in, and measuring how much there was of that mutation in the population as a function of time. And so this, say, curve here, this is, say, one mutation that comes in there, okay? It comes up in the population on some pretty fast time, and then it takes over and it's there in the population. So this mutation arose here and came up and swept through the population, okay? Now you notice these sort of come up in clumps. Initially it goes fast and they slow down a bit, but they come in these clumps. It turns out that's a theoretical prediction. Okay? One of the amazing things about this experiment, this is 25 years worth of data. Okay? At the time when Rich started this experiment, he couldn't have even dreamed of the things being done that I can do. Even the time when I met him, which was around in here, most of the things like this you still couldn't imagine doing. Okay? Maybe you could dream, but you couldn't actually imagine. Okay? And now the rate of development has been incredibly fast. Okay? This basically is consistent with the theory. There are some things that may not work quite, but basically this is what the theory would predict. What is going on here? There's this complicated mess. Something's going up, coming back down again. What that is actually ecology developing. Rich thought this was an ideal system with no ecology, but in fact, the bacteria start making ecology. They start making something interesting. What about out here? Well, rather surprisingly, the rate for things coming in and how fast they come up doesn't seem to have slowed down. The mutations accumulate in what's this nominally constant environment. So this is a really strange result. They're sort of maybe changing things, but it's something which I'm just going to come back and mention something about. Okay. So this is a situation where we tried to not have any ecology, but ecology developed anyway. Okay. So in general, that's something which you have to think about in a serious way, and is absolutely crucial. We have some population of organisms here. We have some mutations. They give then, because of have a higher fitness, they can change the population. They change the organisms. The population will get some of these mutants. The selection acts give the population dynamics, and I get a modified population. So that's all the bits we've got so far. That's the mutations and the selection. But then what does this modified population do? Well, it changes the environment. Right? The bacteria are very good at chemistry. They change the chemistry of the environment. The new chemistry environment affects the organisms. It affects, they're now in a different environment. Different mutations can be good. Okay, other ones that weren't good before can start doing better again, and you go around this cycle. Okay, so this is something which in general always occurs, and then we can ask a question: if I have sufficiently complex organisms and environment, is it possible that the organisms and ecological evolution can continue forever, even under sort of pretty simple circumstances? Like in principle, maybe things you could even do in the lab. Another question is, once you get this, do you tend to always get extensive diversity, a lot of diversity coming up? So you can start getting things here that can coexist with each other. They're all evolving. They're co-evolving with each other, complicated things going on. Does this process, generally give rise to these? Okay. These are extremely big questions, even for these very simple short-term evolution of microbes in simple situations. They're ones that are starting to have some theoretical progress, or at least ideas of theory. I want to mention one of those. Okay. What about what one can see in the lab? Okay, so far I showed experiments which were intended to be pretty boring. Rich Lenski's was much more interesting than he thought it was going to be because of this ecology developing. What about if you don't try to be boring? Okay, so this was a really nice experiment done by Finkel and Coulter some time ago. They just took E. coli, sealed it up in a tube, they shook it so it couldn't develop spatial structure. They had some oxygen and water that it could come in, but it was basically starved. So they expected everything to die quickly. Most of the things did die, but then the other E. coli evolved to be able to eat off the remains of the ones that were dead. Okay? They found in a period of days, amazing set of things happening. They got things taking over, they changed the environment, they learned to eat new things, they radiated a lot of diversity, developed the ecology. And this is a sort of characterization of this, where they would take out individuals from the populations, they would plate them out, they would go colonies, they would look which colonies were different, call that the color in these wedges, and they would just see that would gradually evolve. This is a very primitive experiment, this way of seeing what's there. Right? Nowadays, one could go in and you just sequence from these populations, and you start figuring out what's there. And you can guarantee there's a lot more diversity there. There's a lot more that'll be interesting diversity, not just small genetic changes that don't matter. And one should really be able to follow this. Now, people are going back and trying to start redoing something like this, and also I think apparently some of the samples may be in good enough form to actually be able to look at. But this is saying that even in a simple circumstance, E. coli is a particularly boring bacteria, and in situations where you're not giving it lots of different things to eat initially, it's finding them from each other, you can get all the basic processes of the evolution occurring. Okay. So this is something which is sort of a hint that one might get this process tending to continue. Okay. But how do we, as a theorist, begin to approach that? We can't anymore take, okay, here's a fitness landscape, and we just look at how things move in a population moves in a fitness landscape. Because now we have to say, well, okay, the phenotype, right? The evolution is acting on the is acting on the phenotype, right? It's the properties of the organisms which are coupling to the environment. They're the ones that's modifying the environment and giving you this, right? So you need to think about the properties of the organism and how they're coupling to the environment. So it's not actually that useful to think in sort of DNA space. You really want to think of properties of the organisms. Okay? So how can we sort of can we model those in a way that's even useful at all? Or even that I would convince myself is useful. Whether I can convince you is a different matter. Okay. So, how might we try to simplify things? Bacteria, there's a huge number of properties that determine how they respond to the environment. They interact via a large number of chemicals, and how they affect the environment. Right? They take up chemicals, they change them, they give off chemicals, and so on. So I just sort of list the total number of things here. They're very, very large. Then we can sort of ask about the nature of the biology. We somehow have to have something about the biology that's going to go in there at some point. If we go from molecular to cell biology, the standard picture people have developed of this is that sort of things are rather modular. This gene does this, this gene does that. Okay. However, that's a big oversimplification. Overall it's sort of a mess of interacting parts. Some of them functional well together, but they also have a lot of accidental interactions that are just there because of historical past. Okay. So there's sort of this mess there. And what does that suggest? That suggests that the complexities of the biology, the ecology, and the evolutionary history, right? There's a lot of things that come there that way because of the evolutionary history, including accidents. Okay? What does that mean? It means if I look at any effects of changes, they're going to be sums of positive and negative contributions. Something is better in one way, is worse in another way. Okay? Something becomes fitter in one environment, it's slightly less fit in another environment. Which chemicals emits are somewhat different. So this suggests that a lot of the things that come in short-term evolution, that all of the effects of them are going to be sums of positive and negative effects. How the growth rates change, how they change the environment, what that does to the others, and so on. Okay. Well, once things are sums of positive and negative effects, it's very hard to predict whether they're positive or negative, the sum. Right? And in fact, the experiments suggest that. It really is that it seems that predicting whether given mutations are going to be beneficial or not, even in simple situations where people think they understand the organisms, is basically impossible. Okay? Okay, so we're going to try to use this fact that this is unpredictable, sums of positive and negative things, the fact that there are huge numbers of them, and say, okay, those are the kind of things sort of statistical physicists like, and we're going to try to use that. Okay. So the toy models. Now, what do I mean by a toy model? The model of the fitness staircase, that started out as a toy model. But then we could go from that toy model to a somewhat more realistic model where I had a fitness landscape in a constant environment. I had mutations of different effects, I had deleterious mutations and so on. We could sort of go to a model that got more and more realistic. Okay. What I'm going to do now is really a toy. It's something that I have no claims of anything being connected to anything, but I hope it sort of gives one a flavor, okay, and give a way of thinking about things. Okay. So what are crucial parts? Well, this should be high dimensional. What do I mean by that? There's a very, very large number of parameters that determine the properties of organisms and the environment. Okay. And then the key caricature is that I'm going to approximate the complexities by randomness. I can say if I've got a lot of things that are tons of positive and negative parts, the sum total of them is going to have a lot of aspects that are going to be random. I, of course, have to talk about the statistical properties, say what do I mean by random, but I have a basic a sort of underlying approximation here that at least for these short-term changes and so on, that I can treat it in a way as a first starting point of assuming that it's random. So this has a tradition in physics going back to understanding the physics of nuclei. The quantum mechanics of nuclei is Wigner realized that there was such a complicated mess that it was a good starting point to just assume that the eigenvalues, the energy levels were random. Okay. And there's a huge field that's developed from that in physics and gone to lots of interesting areas. And so this is an approach there, and it's going to be, again, a very good starting point. We don't know yet if it's true in biology. Okay. So the particular model which I want to mention, I'm not going to go through this in detail. I'm going to just say the flavor of the model. Okay. So I call this a fitness snowscape model. So I have a fitness landscape, which I'm now thinking of this as a function of the phenotype, the organismic properties, and the environment. It's very high dimensional, and one's intuition about landscapes are all low dimensional. You know, you draw contour maps in 2 dimensions. Some people can imagine 3-dimensional landscapes. I can't. Okay? But by the time you get to a 1,000,000-dimensional landscape, it's not, you know, how does one understand it? And one's intuition is just wrong. In particular, intuition that you kind of go and you get to peaks, fitness peaks. That isn't what happens. Okay? Okay. This is going to be, the random is highly rugged. It's going to have lots of peaks, lots of saddle points. Right? But I'm then going to consider very simple evolution in this landscape. I'm going to have one type. It's going to get mutations and it'll go uphill. Okay, so it gets a mutation. The mutation is slightly fitter. It'll take over the population. So it's like the one step at a time. But I'm thinking of going up in this sort of continuous landscape. So I'm going by a little bit. The mutant takes over the population. However, then that mutant will cause a slight change in the environment. Right? So there's a slight change in the environment because the mutant is a little bit different. So once it's taken over, the environment has changed a little bit. So the new mutants will see a slightly different landscape, a new mutant on top of this. Okay. So why do I call this fitness snowscape? Well, this is like trying to go hiking in the mountains, right? You take steps upwards, and sometimes it slides, and the snow slides. Occasionally, you may even get an avalanche. And you think you're keeping going up. You're always doing better in your sense of feet going up. You're never really getting anywhere, right? You're changing it as you're going along. So this is really analogous to this picture here. Okay. So the result is that arbitrarily small feedback gives you in a phase in sort of a phase transition sense, I would call a red queen phase, which evolves forever without slowing down. Okay. Why is this called red queen? Well, after Alice in Wonderland, where the red queen, you have to run very fast just to stay still. Right? I mean, if you're on a snow slope or a rock slope is actually sometimes more, it's like you have to go go fast just to not go slide down at all. Okay. What do I mean the not slowing down? I mean the mutations will come in and take over with a rate that initially slows down and then sort of saturates and it just keeps going. And all of this will just wander around chaotically in this space. Okay? And in high dimensions, that happens, say, with arbitrarily weak feedback. So this sort of suggests that maybe the idea of that the evolution can, coupled with the ecology, can give you something that continues forever is a very reasonable one. In fact, maybe in some sense it's really a thing one should just expect. Okay. I wish I could say things about the diversity. I tried to say some things yesterday, but we really don't understand as much, almost anything there. And this is, I say, this is very recent. Okay. So before concluding, I want to say something on just a completely different aspect. Everything I've said so far is for asexual populations. Bacteria until recently were thought to be mostly asexual, and now every time you look more, they seem to be having more sex, even though they're hard to see them actually do it, but the evidence is very strong. And the whole issue of the role of sex and things in evolution, let's say, is very poorly understood. Okay? So a key question, why is sex so popular? It's expensive, you can actually measure some of the machinery cost, but the more to the point is the huge disadvantage of males. Right? If each female can have 2 offspring, then if there's half the population's males, that'll keep her descendants will be roughly the same at each generation. However, if there were no males reproducing asexually, population would double each generation. Okay? Having a small number of males, if you need them for insemination, then you would do almost as well as this. Okay? So why are the 50% males? Well, this is a classic argument that goes back as to that once you've got the stupid system with the sex, then you will tend to evolve towards 50-50. Okay? So there's something you're sort of stuck with the system, and then you get to 50-50, and there's a huge disadvantage because another slightly different type that came in, which had 99% females, would do much better for the short term at least. Okay, so there's got to be some big advantages. Okay? Speeding up evolution and diversification, those sort of classic things, other things are said as well. Maintenance of variation for selection to act on. Okay, that's what people say, but then you have to ask, okay, does it actually, if you try to work out math and things, is that actually right? One thing which is, I think one of the things most said is bringing together favorable combinations. One organism has one combination, this one has another. You bring those genes together, they do better. However, sex also breaks up good combinations. Right? There's some, apparently, there was a study done in Iceland a long time ago of looking at who it pays to marry as far as health of and number of offspring and grandchildren. And so, of course, inbreeding is bad. So marrying first cousins, siblings, certainly bad. First cousins is not good. But what they found is there seemed to be some peak around second or third cousins, that there were some drawbacks of marrying ones far away. Now, it's probably there's not enough other things that controlled are for, and apparently people haven't looked for this effect systematically in organisms you can really control, or at least I was told that they haven't. I don't know if they may have. Okay, so there's clearly some trade-offs here, and this is really then a quantitative issues. It also depends on the time scale. Are we talking about good for the short term or good for the long term? Some lizards, they become asexual. They just reproduce parthenogenetically. And for a while they do well, but then they don't seem to leave any long-term descendants. Right? All the major groups of life that last for a long time all have some kinds of mating. Okay. Also, these effects here will determine how much sort of selection acts on genes, which is sort of how in my one biology class I did when I was 14 or something, terrible biology class, when we learn, selection on genes, or, is it selection on genomes? Well, until recently, one would have learned that bacteria always select on genomes because they don't have much sex. But if they're moving it around, then maybe that's selecting on genes as well. And what's the combination in here? Okay. And the answer is surely that when it's a mixture of those, but what do you even mean by that, right? As to how things are selecting for that. And so these are all quantitative things, right? Small amounts of sex can definitely be good. Large amounts, you start breaking up good combinations too much and is bad. Okay. In bacteria, they only have sex occasionally. They mostly will do it by sort of transferring little bits of segments from one to the other. So this is the case. I've got good mutation there, good mutation there. I transfer this and I combine them. Okay. So at some rate. Okay. So I gave a talk in Hong Kong under the title, How Much Sex is Enough? And it was at an obscure conference at an obscure university, and it made the city newspaper. So this is, sometimes if you want to get audiences, you should give good titles. This one I wanted to be a bit broader, so I didn't give that, and so you were kind enough to come anyway. Okay. So one can ask is, okay, if sex is good for bringing these things together, and certainly small amounts does have benefits. The effect of bringing good things together is more than the effect of breaking things apart. Okay. So in bacteria, this number is very small, but their population is also very big. So we want to ask a question which is a bit like the one we were talking about with the staircase with big populations. What sort of combinations of things come in to determine how fast things can evolve? Okay. And so the result is that as the population gets bigger, the amount of recombination you need, of gene transfer you need, goes down rapidly. So if you're a very big population, only tiny amounts. For those mathematically inclined, the speed up is just some function of the ratios of log R to log n. So again, we get these sort of logs coming in. Okay. So for example, if R is of order n to the minus a third, you can already double the speed in simple models. Okay. So what are n to the minus a third? Well, if n is of order 10 to the 12th, okay, so then an R of 10 to the minus 4, it's not very often, you don't see that, would already have a substantial, have a big effect. Okay. But in general, understanding the dynamics, the effects of the sex, understanding the diversity and so on is very difficult. Okay, and it's very difficult even in the simplest models, like the sexual analog of the staircase model. Okay. So I now just want to conclude with sort of something about where we are in sort of future prospects for microbial evolutionary dynamics. Okay, and I'm really, if I can sort of understand shortish-term things with microbes, I'll feel I at least have made some progress. And of course, I would like to understand much bigger picture things, but I don't even know how to really get going on those. Okay. So we can look at natural communities. And one of the advantages now of all the sequencing things is we can really probe them and we can look at dynamics there. We can look at how things move around. We can look at the statistics of the diversity. It's much harder to get the phenotypes and harder to get the ecology, but we can start trying to do some of that. Within humans, we can look at the coevolution of pathogens in the immune system. And this has already been done for HIV, where you look at the immune system, which works by evolving. You evolve antibodies. If they're selected for by working well, then you get new antibodies and they compete against the immune system. Sorry, against the pathogen which is evolving. Okay. So that one can do. And now also the looking at cancer in humans as well. Okay. Experiments. Well, one can try to look for new functions, eating new things. Switches, spontaneous switching between one state. Oscillators, people have seen these. Okay. The multi-cell development. Okay, that's obviously would be a much more exciting thing. Evolution of ecology. This one, there are some tricks that Michael Desai's developed recently, where it looks as if one might be able to see it developing and evolving, even if you don't know what on earth's going on. Okay, this is by tricks of barcoding, where you literally attach a DNA barcode to every cell. Each cell is its own. All its descendants have the same barcode. Okay, so this is, let's say, the most boring experiment that has been done now, is by barcoding 1 million yeast cells and looking at their descendants. So as they picked up mutations, some of them started doing well and coming up, and you could measure that and measure information about the mutations and how many there were, how big their fitness advantages are, and so on. Okay. The problem is, after a while, the best one sort of takes over. So you want to be able to keep going, and what you do is then you add new barcodes and you sort of keep going. So this suggests that one might be able to actually follow the evolution of ecology in the lab. Okay. The other one which you can do is there's a lot of experiments in parallel, and there's beautiful machines that have been made by Paul Rainey and collaborators now. We have lots of little tiny droplets, and you can make many different communities in each of those. Okay. You can also do something, and this people haven't really done much of, is you can boost evolution. You can say, look, I'm happy to do like I did with crops, and to make things go much faster, I can select on single cells. And I can select for something which has nothing to do with growth rate. I can say, geez, this cell is doing something interesting and weird. Let me pull that out and call that fitter by my definition of fitter. Okay, very strong selection. Okay. You can combine artificial and natural one and try to estimate rates of rare processes. And this people haven't done. They've done things that would sort of enable one to start making estimates. Okay. Theory, posing questions. That's the most important part in some ways. For both experiments and for environmental studies. One wants to know what's useful data to try to take, what's informative. Or given some data, what else would be useful to take? Which experiments to do, how to understand those. Abstract and at least semi-realistic models, I've talked about these. For the ones that are more realistic, one can try to do some quantitative extrapolation. The really abstract models that are sort of understand qualitative effects. So one of the reasons for doing quantitative analysis and quantitative modeling is to really gain qualitative understanding. But in the end, the understanding is a thing that's much more qualitative, but often you have to do the quantitative things to understand that. Okay, and the benefits of sex is certainly one like that. You can wave your hands and say, oh, it's good for this, it's bad for that, and so on. But unless you've got some quantitative understanding of it, you don't really understand it qualitatively either. Okay. And of course, one would like to have some general principles, right, beyond the ones that evolutionary biologists already know. Somehow dealing with the multiple levels and the high complexity, that's obviously is crucial. Okay. So what we'd really like to do is make evolutionary biology more like condensed matter physics. Okay? So roughly speaking, evolutionary biology has two parts. There's the traditional evolutionary biology dealing with fossils and existing species. That's sort of like doing geophysics. I used to do geophysics, some work on earthquakes. It was a lot of fun, and you could do it predictive theories. Okay? Then there is small-scale evolution, population genetics. Individual mutations, statistics of neutral mutations, and so on. That's sort of like doing atomic physics, molecules and things like that. Okay? But in evolution, there's almost nothing in between. And in physics, we know there's this huge area of condensed matter physics, and to get from atoms up to geophysics, you have to build up multiple layers in between. Okay? So the hope is we've started to have little bits of steps on that, away from sort of the atoms, getting towards something which is a bit more complicated, but still very simple. And that's sort of hopefully developing a lot of those areas in between. Okay, but a combination of the experiments, of theory, and of looking at natural observations. Okay. So far, everything I've talked about as far as actual evolution has been seen, and experiments and so on, is all boring. It's eating a bit different stuff, it's growing a bit faster, and so on. Okay. So I just want on my last slide show one other experiment of evolving something different. Because I already mentioned these ones, and some points that will come from that. Okay. So Mike Chandrasekhar and collaborators, they tried to evolve for yeast to aggregate, to stick together. And they did this in a way that's not a very natural selection, but doing in the lab. So they looked at sedimenting. They were sinking down. And if they stuck together, they would sediment faster, so they'd sort of skim them off the bottom. Then they'd put them in growth medium, they would grow up. So they had to be able to grow fast, and they would keep cycling. Okay? And what they found, that quite quickly they got ones here, where here's a clump of the yeast, and they started going, the shape sort of started changing, and they got this here. So they found they could very quickly evolve these yeasts that were stuck together. Okay? But that turned out was not what they'd evolved. What that evolved was a simple form of multicellular yeast. It didn't do what they wanted it to do. It did something different, but much more interesting. It would grow out from a single one here, and instead of dividing, it wouldn't. It would keep growing in this sort of branched way. Now, the problem is if it kept growing all like that, the stuff in the middle would get starved, wouldn't have enough oxygen, so it would do worse when against in growth. So then what happened is it would just divide by fragmentation, and then grow up again. And there's a characteristic size of it growing when it divided, and that characteristic size would depend on how long you sedimented for versus the growth part. So you could select for characteristic sizes. So they found this evolved very fast. This is a multicellular thing. This is the kind of experiment result, which if you ask people who thought evolution experiments were boring, I know because I have done this, I said, okay, which things would be interesting? Okay, and you get roughly the same phenomena as if you think about which kinds of intelligence are unique to humans. People will say certain things, and then they find that a chimp does that. And they say, oh no, that's not what we really meant. This would be interesting. And then they find that, okay, a raven does that. Okay? And the sort of bar keeps moving. Okay? Multicellular development, even of a simple kind, is definitely in the category where people have said, oh yeah, that would be impressed if that happens. But then, well, that's not interesting. Yeast is related to multicellular organisms and so on. But there's a much more important lesson here than that. Okay? The more important lesson is that they got something completely different from what they expected. Okay? And this was already seen in some of the earliest evolution experiments, which were carried out by Leo Szilard. He's a physicist who was the first one to realize that you can make an atomic bomb. And he was doing evolution in a chemostat of where you're growing the bacteria, you're feeding in food, you're draining them out. So the ones that grow faster do better. So it's a nice experiment. But that's not what he found at all. What he found is that they evolved to stick to the walls, so they didn't get flushed out. So they did something different. Okay? That's what happens here. Okay? So there's a very nice story, which which I think is a very good metaphor for how evolution works in some sense. And this is particularly aimed for the physicists in the audience, but I think it'll be understood more generally. Okay. So the tradition in some physics departments of asking at the qualifying exam, or sometimes even at thesis defense, some general question. So this student was asked, how would you measure the height of a building with a barometer? Okay. So he said, I would throw it off the roof and measure how long it took to get to the ground, which is, it's a perfectly good answer. And he was failed, but the professor realized that was being kind of unfair. So he asked one of his colleagues if she could re-examine the student, and she did, and then wrote the story down. So it's actually based on a true story. Okay. So she asked him, well, what other way would you do? He said, well, I would go up the staircase and use the barometer as a ruler. So if you didn't have access to the staircase, well, then I would measure the length of the shadow of the barometer in the building, and I would get it from that. And so she said, what about something that uses some more interesting physics? She said, I would make a really good pendulum, and I would measure the period of the pendulum at the bottom of the building and the top, and the gravitational acceleration is a little bit different, and I would get it from that. So she said, what about something that uses its value as a barometer? So he said, well, I would go to the superintendent of the building, and I would say, if you tell me how tall this building is, I'll give you this really nice barometer. Okay? So at this point, she says, okay, you pass, but as they're leaving the room, she goes, well, but surely you know what the right answer is. And he goes, of course I know the answer you want, but I see no reason why I should give it, right? And so the message of evolution really is a very important one, right? Because you may think you're pushing it in a certain way for it to get fitter that way. It doesn't know what the rules are. It's going to do something completely different, okay? And the fact that there are so many different things that organisms could do to become better and outcompete ones that are there, or moving someone else and doing better there, that's an absolutely essential part of evolution. There's some of these enormous number of different things that could be done. How does one quantify that or sort of model it? I have sort of no idea, but it's really an important lesson to go away with. And another one is that you can actually evolve pretty sophisticated things in short time. So I hope I've convinced you at least that there are a lot of questions which need asking, say tiny bits of progress on some of the boring ones, but they're very interesting and challenging problems for, and I think also very good ones for bringing together biologists and theoretical physicists. Thank you.
So thank you, Daniel, for a very exciting lecture. So we'll have some questions now. So there's time for a few questions.
Hi.
Oh.
You mentioned about randomness, actually, to be able to model some of these
Yes,
phenomena.
yeah.
But do you have any idea about the measures of randomness? Like Wigner had an idea about measures of randomness.
Yeah, so, okay, since the answer, I can just take a random Gaussian manifold in high dimensions and work on that. That's a good starting point, yeah. So it's you basically want something with lots of peaks and valleys and things. So it's
You touched upon sexual versus asexual organisms, but have people thought about why two sexes? Why not more like mating subtypes in yeast?
Yeah.
Thought about that?
I guess there are some organisms apparently that have multiple types. They're rather anomalous. So I think this is one of those things where for the organisms that have it, this can be benefit and once they've already got it. But somehow I don't think it's sort of essential for evolution that the sex and exchange of DNA is somehow is really essential. Okay? People often ask about epistasis, about epigenetic evolution as well. And they say, oh, this is exciting, epigenetic. I say, well, epigenetics is nice, but I sort of don't feel it's essential for evolution. There's other things that can somehow work there. So I think there's probably, and there are examples that have more mating types, but there are sort of oddities, and the general rule in biology is there is always some exception. But I sort of don't, it's not clear why there would be a substantial advantage over just having two, or like bacteria where you just exchange, but you don't have clear sexes.
Can consciousness be seen as a mutation, sort of?
So consciousness Well, to the same extent one can define it all as a sort of emergent phenomena of neurons, I mean, this is not unrelated to the comment about higher intelligence. You know, once you've got sufficient complexity of the way the neurons are wired in the brain, it may be various things just tend to happen because of this sort of complexity of that. And then, you know, some part of that is sort of one thinks of as being conscious and whatever that is, and other parts of it are what enable us to do somewhat more than a chimpanzee, but I don't think there's a sharp distinction there, and I think consciousness is one of those ideas that there's never going to be an answer saying, oh, this is what consciousness is. I think it's...
Any more questions? Okay.
Yeah. So if a species is undergoing a mutation, like it's like a positive mutation.
Yeah.
So you said it will be in so it will be in a step progress, right? So any mutation, like it will. One mutation is occurring in the species. It's progressive in a progressive manner. So it is going to a even more fitter stage in the same landscape. Even more, the species is even more getting fitter to the environment. So will the next mutation will it be occurring at the same time period, or will it be in a very slower rate?
I'm not quite sure I understand what you mean. So the individuals will have get 2 mutations, 3 mutations, and so on, the same individual, their descendants, while there are still other ones in the population. So you'll have a whole mixture of ones with different numbers of mutation. That was like this picture I tried to show about, you know, different steps of the staircase. And actually each step of the staircase, there can be many different mutations that are the ones that actually have that same fitness. So I think you probably didn't answer.
As the mutations accumulate,
Yeah.
does the evolution slow down?
Yeah.
That's the
okay.
question.
Okay. So the general belief is that the evolution slows down and you sort of go to a fitness peak in that environment. Okay. So there are 2 thoughts on that. One is by the time you say it's this very high-dimensional, you know, phenotype space, it actually isn't really true that you go to a fitness peak. You just keep wandering around. You do slow down, but you don't sort of ever get to peaks. Okay. So it's already a bit different than one sort of intuition would suggest. But then this result, which I obviously went by very fast, and this sort of Red Queen result, is that all it needs is for it slightly to change the environment as it's evolving in order for other mutations to be possible to make it keep going without slowing down. Okay? So it's not getting any fitter in the sense if I take one from 100 years from now that kept going and competed against one from now, I don't know which one's going to win. Right? It also will depend on who made the environment, right? Who got there first. Right? And but they all think they're doing better and better. Okay? Now, in some sense, things like exponentially growing populations, like Prochlorococcus floating around the ocean, they can't keep growing faster and faster, right? And so there has to be something else which is going, which is going on. But they definitely are evolving. Right? So it's so there's sort of paradoxes there, and that one really has to crucially think about how they're changing things, how other bits of the environment and things are changing. So,
initially, you talked about rate of evolution,
yes.
estimating rate of evolution. I found that a little bit curious because the way you defined it was only through new mutations. But the way we define evolution itself, which is change in allele frequencies,
right.
that can happen as fast as one cold night in a temperate area, right, where 90% of the population dies
right.
because it's just not
right.
able to survive that cold night.
Right. So you can have very strong selection.
Really
right.
So that it can be very rapid.
Right.
So if you bring in this kind of considerations,
right.
where it's not only new mutations but also allele frequency changes
right.
in other ways, are there any generalities that you can draw?
So then you're talking about sexual populations, right? When you're talking about allele frequencies.
Bacteria, right?
Sorry?
It can be anything. It can be
Yeah.
any organism, sexual or asexual.
Yeah. So in bacteria, if you have extremely strong selection, one of them can just wipe out all the diversity very quickly, right? And with some drug-resistant things, you can get that, right? One just takes over, boom, that's it. Okay. And then you have to get new mutations to keep going. So you can have situations where you just get very strong selection. The strong selection is not likely to keep on going, right? After you've done that, there's not going to be as big as many routes to keeping doing much better. But no, so I was looking at the cases where it's slow, but where you can sort of accumulate a lot of things. In a certain sense, the one big steps are easier to think about, and they're easier to do measurements on and so on. But they're even for antibiotic resistance. What you see in the lab is sort of one big step. What you can see in the populations is not that. It's things that actually involve a lot of steps. And they can be very different, and which ways it happens and things is also different.
So what I'm getting from that is that it can vary based on what kind of evolution you're looking at, what mechanisms you're looking at.
Yeah.
Which is, I think, sort of what most biologists think about.
Yeah. But what one means by what mechanisms is often very unclear, right? And I think that's part of this thing about the complexities and sums of positive and negative effects. It's really hard to say, even in short-term evolution experiments where you have one mutation coming in, as to what it is, why that's actually beneficial, that mutation. Okay, and so that's and typically you change conditions slightly and it isn't anymore, right? So it's the sort of mechanisms, even in simple situations, is not quite clear what it means. Even.
Good evening, sir. Thank you for the talk. Is there any quantitative parameter which tells you how, the degree of ecological niches an organism should be well suited to in order to, that's a measure of its fitness. Say you have certain really resistant organisms which do very well in harsh environments. Say you have tardigrades or some very primitive bacteria. So would you really call them fit in the sense that they're not really well accustomed to environments outside of theirs, and also that their genetic makeup is such that they're really resistant to mutations also?
I mean, this is why I don't like the word fitness, right? Because it's something that grows, really slowly in some particular place. How do I compare that with something else, or I take the same organism and move it somewhere else?
Exactly.
Right?
So do you have a general definition of fitness?
No. And I think trying to make general definitions is a very bad direction to go. And people like to do that in evolution, and they argue about what does group selection mean and multi-level selection, and one can get caught in all kinds of things of thinking about definitions. And one is better to think about some particular phenomenon in some context, and then you can ask, okay, can I generalize a bit in thinking about that? Right? But one wants to avoid being stuck with definitions, and I think that has held back the field actually a lot. And even, I say, thinking of fitness as sort of being one thing, your organisms, if they do badly in that environment, but they survive, while some other organism may do much better in that environment, but the environment changes something and they go extinct, which is fitter, right? And so you're depending on the time scale there, so which is important. And so when all these things, one has to think about those things going on. And of course, if you try to do everything at once, it's too complicated to work through anything quantitatively or often even qualitatively, and you can sort of divide the parts up. But one has to have in the back of one mind the thought that, okay, one is only thinking about one part of it, and there are going to be other parts that coming in and crucial things as well, and not try to sort of impose, sort of one size fits all on questions like that.
So can we have some questions from somebody who doesn't have a Y chromosome?
Yeah.
Women, there's so many women.
Yeah.
Come on, ask a question, no? No? Okay.
No. It's someone who's less than half my age.
Yeah, that's fair, yeah.
Yeah.
Am I audible to you? Yeah. In one of your slides, you showed that for a very huge population of bacteria,
Yes.
as n increases,
Yeah.
the rate of transfer of segments, HGT, it decreases.
Yeah.
No. Is that a biological thing, or is it just because the population size is now?
No, sorry, I said that in a confusing way. So the the rate at which you get gene transfer depends a lot on the conditions of the environment and is not much as understood. What I meant is that if you have a, if you ask how much gain does the population get in how fast it can evolve by having a gene exchange, gene transfer, okay, then if you have a population which is a very, very big population, then the amount of gene transfer you need to gain from that is really, really tiny. If you have a modest-sized population, you need more in order to gain, okay? So it's not how much actually takes place, it's how much benefit is from that. So with a very big population, I mean populations like you have in your gut, you know, if even E. coli you've got 10 to the 11 or something, of that, by the time you get that big a population, the amounts of recombination that you would see that would really make a big difference already are something which is hard to actually go and measure. Right? I mean, it's tricks you can if you include it with selection effects. But so it's an effect about how much recombination will help the population evolve, which depends on its size. Yeah, that also will depend on the environment. Yeah. But how much recombination actually occurs is a different question. That of course can be under selection, and so the organisms can somehow evolve to have a better amount of gene transfer, and that certainly happens. I mean that in some environments, organisms certainly exchange DNA much more often than others.
Hello. Right. Yeah.
Well, you have a Y chromosome though. Is there some?
Yeah. Sorry for that.
Oh.
So the thing is, you said that if we place some organisms in a certain landscape, and in order for their survival, they undergo some certain mutation and they thrive.
Right.
Now, because of some change in landscape, that certain mutation actually becomes a negative factor for their survival.
Right.
Now, can you predict that the next mutation will be the reverse of this mutation?
Okay. So you can almost surely predict it won't be. Okay. So then this is one of those ones about number of ways of doing things, right? If it's exactly the same mutation to reverse, for E. coli, say, the chance that is 10 to the minus 10, exactly the same ones. But typically there'll be many others that can compensate for some of the bad effects of the first one. So this is in fact exactly what happens with antibiotic resistance, is you have an antibiotic that gets some mutation to do better with the antibiotic. Then you take the antibiotic away, and it can't compete with the other ones. So you say, okay, well, what's going to happen is it's just going to revert and get the reverse mutation and go back. That's not what happens. It gets some other mutations, compensatory mutations, and there are many possible ones, so it can get one of them easily, even though because there are many possible, and then it can do better. Okay, and that's the sort of crucial process by which bacteria become antibiotic resistant, become problematic, is when they can do well without the antibiotic there. So they.
Hi. So I wanted to ask, in the context of microbes, where they quickly divide and also affect their environment, what is the meaning of long-term benefit there?
Did I use the word long-term benefit? The phrase? It depends. Well, so this is one where I can certainly ask about the descendants of a given individual on lots of timescales, right? So I can ask, is one of its lineages that comes from it going to not go extinct, but most of the others go? And in fact, even this Rich-Lenski experiment, in a different run, he had 12 different lines he ran together. In one of them, there's something which is there in tiny amounts in the population for 30,000 generations, and then comes up and starts taking over when the ecology changes. Right? So that's one which wasn't being successful at all, and then things changed a little, and then it was. It sort of evolved more in things. So, but it was sort of very good in the long run, but not in the short run. So it's not. It depends on the. You have to kind of more quantitatively ask the question, and in particular context. But it really is one which is a really crucial feature, is that thinking about the fitness or things like the fitness on many time scales.
So could we say that maintaining heterogeneity in microbial population is sort of a fundamental property of it?
Yeah, I mean, they're big populations, so they always have heterogeneity in. So let me actually I'm keeping going back to antibiotic resistance because it's a nice example. If you take a single bacterium and you grow up a colony from it, Okay. By the time you've grown the colony up, you've already got most of the possible mutations that can occur. So you already have got one in there that's antibiotic resistant. Okay, that's just there because you've got a big enough population. So it's different than with us, where our populations, modest size. Well, now they're big, but they historically they've been relatively modest sized. The bacteria tend to produce quite a lot of diversity as they get it quite quickly. They get most of their interesting diversity from transferring DNA between different ones. So if you put a bacterium that doesn't have antibiotic resistance into a pig's nose, I think this experiment was done, and then you put some other ones that do, and they have it on plasmids, on little rings of DNA. The first time you make the measurement after that, you find some of them, they've already acquired it from it transferring. So then they're getting a whole interesting new function in one go. Okay, so the sort of diversity in the populations, always occurs. It's very hard to find microbial populations that don't have a lot of diversity. But why they have so much within individual species is one of the huge puzzles. So that's.
Okay, thank Daniel once again.